Hi, everyone. Welcome back to the TLA Podcast. Today, we're talking about something small, very small, but extremely powerful. Have you ever wondered how a microscope actually works?
I love that question. We've all seen microscopes in school labs, but most people just adjust the focus and look through the lens without thinking about what's really happening.
Exactly. It almost feels magical. You place something tiny on a slide, look through the eyepiece, and suddenly an invisible world appears.
But it's not magic, it's physics, specifically the behavior of light.
Let's start with the basic idea. A microscope works by using lenses to magnify very small objects. In simple terms, it bends light in a way that makes tiny details appear larger.
Right, the most common type is the optical microscope, which uses visible light. When light passes through or reflects off a sample, it enters the microscope's lenses.
And those lenses are carefully shaped pieces of glass. Their curved surfaces bend the light rays. This bending process is called refraction.
Inside a standard compound microscope, there are two main sets of lenses, the objective lens and the eyepiece lens.
The objective lens is located close to the specimen. It produces the first magnified image.
Then the eyepiece lens magnifies that image again. So what you're seeing isn't the object directly, it's an enlarged image created by two stages of magnification.
That's why it's called a compound microscope. The total magnification is calculated by multiplying the magnification of the objective lens by that of the eyepiece.
For example, if the objective lens magnifies 40 times and the eyepiece lens magnifies 10 times, the total magnification is 400 times.
Which means something extremely small becomes visible in detail.
But magnification alone isn't enough. There's also something called resolution.
Yes, and resolution is just as important. It refers to the microscope's ability to distinguish two very close points as separate.
If the resolution is low, everything looks blurry. If the resolution is high, you can see fine details clearly.
Light microscopes are powerful, but they have limits. Visible light has a certain wavelength, and that wavelength limits how small a detail we can see.
That's where electron microscopes come in. Instead of using light, they use beams of electrons.
Because electrons have much shorter wavelengths than visible light, electron microscopes can achieve far higher resolution.
With electron microscopes, scientists can observe viruses and even the structure of cells at a molecular level.
But they're much more complex and expensive. They require a vacuum environment and specialized equipment.
So in everyday laboratories and classrooms, optical microscopes are still the standard.
Another interesting part of how microscopes work is illumination. The sample needs proper lighting to be visible.
Yes, light usually comes from below the specimen and passes through it. The amount and angle of light can dramatically affect what you see.
That's why microscopes have adjustable diaphragms and light intensity controls.
When you adjust the focus knob, you're actually changing the distance between the objective lens and the specimen.
And that changes where the image forms. When the image aligns correctly with your eye, it appears sharp.
It's a precise balance between light, distance, and lens curvature.
What fascinates me is how much microscopes changed science. Before their invention, people had no idea that cells even existed.
Exactly. In the 17th century, scientists like Robert Hooke and Anthony van Leeuwenhoek used early microscopes to observe tiny structures for the first time.
That discovery completely transformed biology and medicine.
It's amazing to think that something so small, a lens bending light, opened the door to understanding bacteria, cells, and entire biological systems.
And today, microscopes continue to help scientists develop medicines, study diseases, and explore materials at the microscopic level.
So next time you see a microscope, remember, it's not just magnifying, it's manipulating light in a precise and scientific way.
It's revealing details that our eyes simply cannot detect on their own.
A small device, but a revolutionary invention.
Thanks for exploring the microscopic world with us today.
We're really glad you were here.
Stay curious.
And keep looking closer. See you next time on the TLA Podcast.